Hello, my name is Zafaryab Haider. I am presenting on the topic self-supervised extreme compression of gigapixel images. So, whole slide images, also abbreviated as WSI. So, these images are microscopy images of diseased tissue, and these are the images that are routinely taken in the cancer treatment centers. So it is more like the part of the computational pathology involving observation of tissue slides with a microscope, and it is the gold standard for the cancer diagnosis. So in recent years, digital pathology has emerged as a powerful technology for digitizing whole slide images for assessment, sharing, and analysis of the cancer tissue cells. And so they contain complex and abundant information on thousands of individual cells, their environments, and the overall tissue architecture. So the most important task here in the computational pathology is to make predictions directly from the whole slide images, and we will be looking into this shortly. So these whole slide images are extremely large. So if I take a typical sample of 20 by 15 size of image, and if you digitize it with a resolution of 0.25 micrometer per pixel, that is MPP, and now these images are going to be huge images because they can be magnified for up to like 40x times or 20x times, or if you go for a lesser MPP value, then you may get up to like 100x kind of images as well. So assume that the resulting image is about 80,000 across 60,000 pixels. So it will be around 4.8 gigapixels. If it is represented in RPG, so we will have 24-bit color channel as well. So the image size would approximately be about 15 GB. In extreme cases, there could be a sample of like 50 by 25 captured at zero point 0.1 MPP with 10 Z planes, for example, and the size of one of the images is going to be like 375 GB. And the other thing is, with the advancement of the technology today, it is possible to generate images of that quality. So if you keep on zooming onto the images, and you will just get a finer and better resolution detailed version of that image. This becomes a bottleneck. And now this kind of tiled representation has got some issues with the zooming. So the problem is that this high resolution or the small image area must be accessed to render a given region, which is exemplified over here by the dark green image again. So at lower resolution, progressively larger image areas must be accessed to render the same size of region. At the limit, to render a low resolution thumbnail of the entire image, all the data in the image must be accessed and downsampled. And when you're downsampling it, uh, you have to, you know, downsample at that moment, and then you have to render that image. So one of the ways to go about is like the pyramid architecture, and this is what the WSI's whole slide images are stored as. So you have got a base where you have got a detailed picture, and then there are different slides that you form out of it. So you pre-compute the lower resolution version of the image, and there are different low resolution versions. So they are typically spaced like with some power of 2. So it's like if it is represented by 8,000 pixels, the lower one will be represented by like 4,000 pixels, and the lower one by 2,000 pixels. And so by some power of 2, the size keeps on reducing for the lower images, and then at the very last level, you may have a thumbnail representation of that particular region. Any typical organization of a WSI for pathology or the cancer cells that we are talking about is actually stored in this pyramidal shape. So I would like to show you the example of how these images look like. This is not very clear, but it will give you an idea that at the very zoom out level how it looks like, and when you zoom into it, a more refined version of the image is getting created, and you can see at the cellular level what's happening inside. So state-of-the-art approach for the classification of such images is that you subdivide your WSI into tiles, as I said earlier, and then encode them by some pre-trained networks and apply multiple instance learning to train for specific downstream tasks. So This is like a classical binary supervised learning problem that aims in finding a model that predicts a value of a target variable. And the term weakly supervised is very important over here, because you have to understand that when you get the images of such high order, the annotation on the images is one of the tasks that is not easy. And most of the time, you get images that are not annotated, and therefore the topic self-supervised of my presentation. So when imagine you are getting images that are not annotated, so they are very weakly supervised because you don't exactly know what areas to look for and what not. So how does this weakly supervised work is that you have, it's a binary system and you have to predict whether the patch that you are dealing with has the cancer cells or it doesn't have the cancer cells. So just imagine a bag has got many instances. If any one of the instance is positive, then the bag is gonna be positive. If all the instances are negative, you have to label that bag as negative, right? So the crux is, if any one of the instances is positive, that means there is cancer, and then the bag should be labeled as cancerous. Otherwise, it should not be labeled as cancerous. So this is an example for that, where the blue ones are the positive. So even if one is positive, the bag is labeled, should be labeled as positives, right? And for negative instances, no problem, because if you don't have a cancer, there is no worry. So this is your weakly supervised classification, that you don't look into all the instances. Just one instance, if positive, is making is helping you to create that decision boundary in order to label the bags. This is the equation that represents the multiple instance learning, and it is implemented as a combination of three modules. And these three modules are like E theta 1, which is the embedder, then there is a pooling operator P theta 2, and then there is a classifier C theta 3. X1, X2, and up till Xn are the instances that are present inside the bag. So here every instance is first embedded, and these embeddings are then cumulatively taken and aggregated with some pooling functions, and with the pooling functions we decide that what kind of a label should be put onto the bag. So there is this third classifier that basically classifies how to label the bag. Now this pooling mechanism P theta 2 is something that could be implemented in two different ways. One of the ways is like going for the instance as IID. So IID means that it is taking all the instance independently, and taking instances independently means they do not have any relationship between them. And then the other method of creating a polling function is to exploit the relationship between the instances present in a bag. Now these images are huge, and if we are talking about the instances present in these images, that means they are basically belonging to the same image, and they somehow are correlated with each other. So we cannot go for we can go for the IID space, but for Gigapixel images, therefore we should consider the correlations and the morphological data and the spatial data of those cells as well, instead of going for like independent instances, because these are not independent at all. They are dependent on each other or correlated to each other in some way or the other, right? So the creation of this P theta 2 is something that is tricky, and that requires a lot of research into it, and some methods have already been developed. So one such mechanism is implemented over here, where we have got a bag of instances, and these instances are classified using some standard classifier, and based on the score that they get, the highest score is then put forward into another CNN based classifier, where the weights are updated, and the score is calculated based on the probability and the result that the classifier gives you. Now this is based on CNN, so it goes through some shortcoming, like feature extractor could suffer from insufficient training and noisy gradients. So one of the reasons is that we don't have humongous amount of data present for training purposes. So the second approach that could be a little more helpful is that instead of going for just one particular highest score instance, this mechanism outdo some of the problems of the previous one here, you have got an F function that is more like a feature extraction mechanism. So you could use any of the algorithms like ResNets for extracting the feature out of it, and based on the features that you have received, you pay equal weightage to all of them. That means you aggregate them into an aggregate function, and then now this aggregate function learns the bag representation and predicts the bag label. This results in bag embedding, and then bag label prediction is carried out, and the bag label is provided to the bag. The next one is your trans MIL for Google Pixel images. Now, this is something that uh, helps in attaining the morphological and spatial data of the images, and it's more leverage to produce accurate prediction. So modern MIL architecture for Google Pixel images have been designed to exploit this spatial relationship of the instances. These are the transformer layers basically, and then this is a PPEG, then this is another transformer layer. So what happens here is that the image is first of all broken down into the patches, and then these patches are supplied to the pre-trained ResNet 50, from where we get some feature vectors. Once the features have been extracted, they are provided to the squaring function, and which later calls on the transformers. So these transformers are something that are designed for the vision tasks. So they use softmax for it. So it's like a row by row softmax normalization function. So the standard self-attention mechanism requires the calculation of the similarity scores between each pair of tokens that it receives. To deal with these long instances queue problems in WSI, there is a Nystrom method that has been adopted for this particular implementation by the authors, and then it passes through the PPEG module, which uses different size convolutional kernels in the same layer, which can encode the positional information with the different granularity, enabling high adaptability of PPEG. So it basically takes the advantage of the CNN's ability to aggregate context information. So this is one of the ways of going about the performing of MIL for gigapixel images, and this work has been published in 2022 itself. So, and it shows some very promising results. If we talk about self-supervised learning and classical image processing, then the best example here is similarity CLR, which is contrastive learning. So contrastive learning, if you see, is doing a very good job in self-supervised learning, which is almost equivalent to the supervised learning, and this is something developed by Google. So why this name contrastive learning? Because if you see at this diagram, then you have got an image. Out of those images, now these are not labeled images, so you perform some data augmentation. The data augmentation is a transformation of image into like grayscale or cropping or any kind of a transformation that could take place. And once a dog image is getting transformed into two different images, we know that these two images belong to the dog, so they are fed into the CNN, and when their representation is flattened and we receive it, we tend to give a more similarity score if the augmentations are coming from the same image. On the same side, we also go for different images as well and try to create a negative score for the dissimilarity they have. Therefore, the name contrastive came into this, that we are not just giving positive marks for the similarity, we are also giving negative marks for the dissimilarity. So this way we create a divide between the similar kind of features that then club together to form a label for that particular bag. So this loss function will explain more what I was saying, because the numerator over here is similarity of the positive pair. So if I'm talking about I and J, so if this image gets broken into I and J, this is the similarity that I'm calculating, and I'm giving it a high score. And then there is a similarity between I and K. Now K is something else than J, and I know that transformed or augmented data doesn't belong to the original image I that I'm talking about. Therefore, I'll be giving some negative marks to it, and thereby I'll be creating a difference between the plotting of these kind of informations in order to form the cluster of similar type of images together, and the other cluster should be at a distance from the similar cluster. Then there is self-supervised learning for gigapixel images. It is motivated somewhat by the hierarchical representations in the natural language processing, where you can imagine now this big picture as the paragraph. From paragraph you extract sentences, and from sentences you extract words, and from there you extract the letters, and then you try to find the feature in that particular order in order to make your algorithms learn. So here it is more like that only. When we get the cell level aggregations, we pass it through the MLP layers, and then through the VITs to generate the visual tokens, and then these tokens are pulled up and the global pooling is applied, and then you get just one patch level representation that actually represents this complete patch. And similarly, you collect all such patch level representations, again feed it to the same network. It's like pre-patch MLP, and then visual tokens are generated, and then you get one region level representation of this particular patch, and then you collect so many regional representations, and again go through the same process, and then finally you'll get a slide level representation. Think it of this way, that if one of the instances has been identified as cancerous, then it gets replicated back to the larger level, and the whole image is then marked as the cancerous image. So you look into the cell level aggregation, and then you propagate that back to the higher regional level aggregation as well. And the performance of the HIPD. Now, this is something that has been published very recently in 2022, and it has shown some promising results on different kind of data. So the challenges that we have been discussing in this particular presentation is that the WSI images are huge, and it's very difficult to have them at the same time in the GPU and work on them. And the amount of data available as WSI images is not humongous, and the annotation data that we have is also very limited, and therefore it goes through many problems. So one of the proposed solutions is that which is illustrated in this diagram, and this is called as the Giga SSL, or the Giga Self-Supervised Learning. This basically comprises of 5 important components, and these components are the Gigapixel image level augmentation operator that transforms the images. So you'll see here that the image is divided into the tiles, and then two views are formed, and these views, the upper one is called as not shared, because here the transformations of the different tiles are not shared. So one of the slide may go through like a cropping transformation, the other may go through some coloring transformation, some jitter transformation, or anything. And in the next one, here the tile level augmentations, the transformation is applied same to the different tiles that have been selected. And then on the next stage, the slide level augmentations are performed, where the slide as a whole has been transformed into a new representation. This could be through scaling, rotation, or flips, or whatever. Then these are fed into the pre-trained ResNet 18 networks, and here the features are being extracted, and now this F represents the number of features that have been extracted out of the images that have been provided, and T here represents the number of samples that have been provided into it. For example, if we are providing 5 sample tiles into it, or 4 sample tiles into it, so T represents that number. The next matrix over here represents the coordinate, or the actual location of those tiles present on this particular slide. So where these tiles belong to, that information is in this particular matrix. So when these two matrices are multiplied, they are fed into the aggregation model, and this aggregation model is going to be something represented by the pooling P theta, where you calculate the MIL or Covenant or transformer. You can apply any of the algorithms with the weight optimized with the SGD that would result in some data points then which can be calculated through the loss function using any contrastive learning mechanism, which could be SimCLR or MOCO. For this paper, they have used MOCO and they have used ResNet 18. And they have sampled slides of like t equals to 5 per slide, and then there is this bootstrap that I'll discuss later on. Fine. So one of the bottlenecks for this particular instance is that if there are B batches, then two views would be created. So there it will multiply by 2, and then there are 8 sample types that are getting picked up from these samples. So there will be like 8 uh, times, there will be B cross 2 cross T times images available that have to be fed to this pre-trained ResNet 18 network, which is huge. So, and we have got this GPU limitation as well. So this is one of the bottlenecks that this scheme needs to have. If somebody argues that we can reduce the batch size, so we cannot, because the author has mentioned 2 other works of the people who have proved that batch size should be high, because if it is not, then contrastive learning or the self-supervised portion of it will not be efficient enough. Therefore, we need to have a lot of batches. We need to have then from those batches 2 views and T samples. So there's going to be a lot of images. So we need to have something in that regard. To overcome this problem, one of the solutions is the pre-computing augmented representation instead of calculating it online. So if the tile level augmentation are not shared among the views, then the authors have randomly performed some transformations on each of the tiles, and whatever the embeddings they got out of it, they just stored and stacked those embeddings. But if the tile level augmentations were shared, that means they had same kind of transformation across the tiles, then for each image they took out like 50 samples out of it with different augmentation and apply all of these transformation to the batch of like 256 tiles, and randomly sampled in the WSI. So whatever embeddings they got out of these transformation, they were kept in a separate folder. And at the training time, these pre-computed samples were used instead of calculating the samples on the go. So that way, the authors of this paper observed that they did the same task in 10 hours of GPU time, which was actually taking them about 20 times longer to train the models. So that's a significant improvement in the performance just by pre-computing of the augmented task embeddings. Thing important over here is that when you are working on the embeddings, then you not necessarily are working on the huge information that is given in the images itself. Therefore, they have also observed that 16 terabyte of image requirement, which is there in the data set that they were working on, which is Cancer Genome Atlas, that is TCGA. So that actually from 16 terabyte came down to 23 MB of data on which they were working. So it was very efficient in terms of storage as well. So the GPUs were not throttled to a great extent, and the better efficiency were recorded by the authors. The results shown by the authors are very promising as well, because this is the Gigapixel that they have proposed, and for different kind of data sets that they worked on, they almost got better results in every except for RCC subtyping. And the good thing to note over here is that when they were using only 25% of the train sets, still they were getting a very good accuracy as compared to all other people. So it's like that they work better in the tiny data sets as well, which is one of the requirements, as we said earlier as well, that we don't have enough WSI images that are annotated. So we are limited by the data sets, and we need to have mechanisms that can perform self-labeling and perform very well with the tiny data sets that we have. So this work looks quite promising in those regards. Then when it was compared with the deep MIL for the tiny data sets, then again it performed really well. So this red line is basically something that's telling that both the algorithms are performing equal, and anything above the red line is in favor of the GSSL or Gigapixel self-supervised learning, this current paper. So if you look at different data sets and the sizes, you will see that every time, except for a few places like here on CRC dataset and NSCLC dataset, things were like equal, but in all other cases GSSL has performed better. And then sensitivity analysis was also performed by the authors, and they found that for the lesser number of tiles, they were getting a better accuracy with different train sizes. So when the number of tiles was less, their accuracy was better. So fewer tiles, the better. So next one is ensemble size. Ensemble size, it was calculated that how many views of a particular slide can be created with different tile selections in order to have a good embedding towards the end, and it was found that it optimizes somewhere at around 50. So if 50 views are generated from a particular slide, picking out random tiles, then the output comes out to be really nice. And then the third one is the epochs. The weird thing about this third one is that there is a mechanism called as attention deep MIL, which I have not discussed in this particular presentation, but it performs really well with the classical images. But somehow these authors did not report a good performance of this attention based deep MIL with the gigapixel images. So you'll see a downwards trend as the number of epochs keeps on increasing for the attention, but it was working fine with the Sparsian techniques. So this is all from the paper that I presented. Thank you. These are the references from where I read about various concepts. Thank you so much.